Okay, so just an overview of what I'm going to talk about today. I'm going to give a really brief introduction and then just move on into talking about some aspects of pollinator diversity in Southern Illinois. My lab did a large scale inventory of flower visiting insects a number of years ago. And I'm going to kind of carve out some little vignettes from that I thought would fit in with the theme of the symposium, and that highlights some student work that has happened in my lab. And then I'll talk a little bit about pollinator diversity on two military training sites, which is the most recent field project that I have done. So these little sort of stories within a bigger picture will be a look at limestone glade bees from Southern Illinois, as well as host plants of a species of concern, a bumblebee, a look at syrphid flies and their potential as pollinators, and also Lepidoptera and their role as pollinators as well. Okay, figure this out here. So most of you have heard things in the media about pollinators. There's some conservation issues that have been increasing over time, declines in native bees. There are now several bumblebees recognized either through federal or state listing as threatened or endangered. Six indigenous bees from Hawaii have also been added to the Federal Endangered Species Act. There's quite a growing list of bees and other pollinators to be considered for state or federal listing. There have been general pollinator declines and general insect declines that have been detected in North America, in Europe, and in other places. This has been linked to loss of habitat and loss of floral resources, and in particular, native floral resources. And I know I'm sort of preaching to the choir here, but I'll be highlighting the importance of native plants. And then finally, even though honeybees are also claimed erroneously to be in some kind of danger, they're actually one of the stressors on our native pollinators, as are problems arising from other kinds of non-native species, such as invasive weeds. So just a few things that, just some random grabs from news articles here, looking at pollinator losses in Britain, the fluctuating monarch demographics, and some paper, a very interesting paper that I cite a lot over there in the lower corner, that actually tried to measure the impacts of commercial apiaries on native bees, and it wasn't pretty. Not really the topic of my talk today, but all of those things are relevant. There's also been some research published that's relevant to the general. We don't really consider this Southern Illinois, but probably everybody up in Chicago would call Carlinville Southern Illinois. And there's a really important paper that was published in the early 1900s by an amateur naturalist, Charles Robertson, and then where he in detail recorded what plants were present in a prairie area, what bees and other pollinators were visiting, and also which plants they were visiting. And he even made an attempt to determine whether they were feeding on pollen or nectar or both. And then that's been revisited, but in the 70s and then later in the 2000s, where researchers comparing work to Robertson's early data, have found evidence of species loss, potentially declining pollinator services for plants, degradation of pollinator networks. I'll be showing you some pollinator networks, but it's a way to visualize and also do some analyses on the interactions between pollinators and plants. And also, importantly, phenology shifts, things that were active at the same time, plants, flowering plants, and active adult pollinators, for example, being out at the same time where they can potentially visit. If climate change causes the same effect in both of them, they might be okay, but if it speeds up plant phenology but not bee emergence, then there's some problems. Okay, so I'm gonna just kind of go over some general methods that uh, all of the studies that I'm going to talk about kind of follow the same, really the same set of methodology. And so the data sets that I'm going to be talking about are a pollinator inventory that was conducted in 2017 through 2019 in Southern Illinois, with a focus on federal lands because it was a federal grant that was supporting this work. This was done with collaborators Carla Gage here at SIU, also Casey Bryan, who really spearheaded this whole project, who was a grad student at the time, now working for the Fish and Wildlife. And in addition to doing some work with ag fields in Crab Orchard, we also did an ecologically stratified inventory, meaning that we tried to collect pollinators from as many different kinds of habitats as possible, which included ag fields, fallow ag fields or edges of ag fields, open meadows and prairie type locations, glades, forest interiors and forest edges, and plot twist, or maybe spoiler alert, closed canopy forests do not support very many native bees. You need sunshine and sun-loving plants. So lots of roadsides, tried to include wetlands, and some urban collecting as well, some opportunistic collecting. And this also included an intensive study in one of those years of cover crops, weedy plants and pollinators in the ag fields in Crab Orchard. And then 2019 through 21, my lab inventoried two military training sites. And I haven't presented this to anyone, so you guys will be the first ones besides the Army guys I sent the report to hear a little bit about those. So very quickly, our methods. We used established methods for sampling bees and other pollinators, and these included at each site that we visited, both targeted hand collecting as well as using bowl traps, which are basically bowls painted with colors that are in general attractive to insects and filled with some water with a little bit of detergent in it to break the surface tension. And we collected almost all insect visitors that we encountered. I say almost because for conservation purposes, we avoided collecting monarchs and queen bumblebees in early spring and late summer so as not to impact those populations. We had sites that we revisited, as well as a lot of sites that we just called opportunistic roaming, which is basically what it sounds like, driving around the countryside and looking for usually roadside, something in flower, whatever's in flower at that moment. And it's a real moving target and lots of area to cover. So it kept us busy. At each site, we usually did about 80 person minutes of collecting. So that would mean 80 minutes if there was one person there, or 40 minutes if there were two, etc. New people got counted as half a person until we were sure that they'd come up to speed in terms of collecting as much as I could collect, or my seasoned students, such as Jacob here. So that plus the pan traps, which provided a passive hands-free means of augmenting the sampling, increased the number of species that can be detected. Additionally, we had to ID the plants in flower, and we kept track of floral associations, which means when we collected a bee, for example, from a particular plant, we kept it separate with that information of what plant it came from. So our database has floral associations for all of that targeted hand netting, although there are obviously no floral associations for the things that end up in the pan trap. The work doesn't end at all after the field seasons are over. So back in the lab, pinning, labeling, and sorting everything. Large numbers of very small animals present challenges for curation. And basically things have to be kind of moved through some stations and be kept very carefully until everything is pinned and has its own label on it. Then we can start sorting and going through the process of taxonomic identification. Our lab would always identify things to the lowest taxonomic level that we could get it to, and if that was species, then that would be great. We were finished, but frequently we had encountered taxonomically difficult groups that were sent to appropriate experts so that we have accurate species identifications. And all of this is this collection is data-based with all of the associated information with each specimen, in addition to what species it is, the location, when it was collected, and the floral association. You can see some of these very tiny labels here. They're printed in 4-point font on archival paper, and then somebody has to cut them up and put them on the pin. They each get a unique accession number. And I just want to plug undergrad or just student training and mentoring, which there was a lot of in this project. So we had a large group of undergraduates as well as several graduate students working on this. And I'll put anybody to work sorting bees. And no matter how little you know about them, I can teach you a few things and have you helping out with the group. So everyone of it, every experience level helped with this. So there was lots of hands-on training. And then for those students who stuck around long enough and had an interest, training in using dichotomous keys, for example, to identify various taxonomic groups, and general collection curation techniques as well. And some of these students were supported by an NSF training grant that focused on research with collections that Dr. Newbig led for a number of years. So a little bit about our main inventory that took place in 2017 through 19. A fun time was had by all in the field. And here are some looks at that. That was a fun day. That was a fun day. And my personal vehicle became this official looking pollinator inventory research team vehicle. And by the end of these 2 years, everyone, including me, had gotten this truck stuck somewhere because it was 2-wheel drive, but it made you feel like you were in a real truck, but it couldn't get you out of any place like a real truck. So we drove it into the ground, and then it is not with me anymore. Okay, so at the end, by the end of this project, we had our total revisited sites plus the opportunistic roaming sites were numbered 237 with over 1,100 person hours of hand netting and almost 1,200 unique collection events, so place, time combinations. And this is a number that we had at one time. It's not the current one, but it kind of shows you a general idea. I think we're inching up towards 50,000 now. Um, about half of it was collected through the targeted hand netting and half through pan traps. Most of them were bees and other Hymenoptera, Lepidoptera, or butterflies and moths, Coleoptera, the beetles, syrphid flies, and a bunch of other flies too that we are still in the process of working on. The wasps and the other kinds of dipterans were sort of deprioritized a little bit so that we could focus on the groups that are probably the main pollinators. But that being said, I'm really excited to get the wasps done because I think we're going to learn a lot about the role of wasps in pollination in communities. Okay, so at this point, you might be either thinking that's really cool, or you might be completely horrified because we've killed 30,000 pollinators. And so I need to explain why we do this. We do it because if you just collect something until you think that, and then when you think you have something similar looking, you let it go or something, you're going to miss a huge amount of the diversity. And so we can do something called a species area curve where you have the number of species, the cumulative number of species sampled versus some measure of sampling effort. With plants, this is usually the area, total area sampled, but with the kind of collection we're doing here, person hours is the more relevant number. And so you get this curve where as you sample more, you find more stuff up to some certain point. And eventually, it's diminished returns as you grow closer and closer to the real diversity that's in the area. And so you don't want to be right here, and you have to collect a lot to not be there. We're probably here or something. I have no illusions that we collected every kind of pollinator because we didn't. Um, but this happy spot up here, I will say you probably never get there with any kind of invertebrate, certainly not insects. Can never really, unless you're doing some kind of very long-term, well-funded and well-staffed study. So a little bit of kind of a summary of what we found in terms of species numbers, as well as a little bit that give you some numbers to give you a clue about how these are interacting with plants. So 239 species of bees, I think, is close to our current number. And most of these are native. There's, in addition to honeybees, which are non-native, there's a handful of other bees that have been inadvertently introduced and are not native, but they're also not really invasive. They're probably not doing a whole lot significant. Honeybees are significant, but again, not the topic today. These had we found these, we collected these from over 300 plant species. And then the unique visitor flower associations are simply the number of different bee plant or visitor plant interactions. 139 species of lepidopterans, and that's only from diurnal collections. We didn't collect at night, so that doesn't include any nocturnally active moths, which were collected from over 200 floral associates, and from which we recognized 751 unique visitor flower associations. 69 species of syrphids, 64 flower visiting beetles, for a total of over 500 flower visitors. And again, with the caveat that the wasps and the remainder of the flies are not being shown here. They're still being worked on. These included at least 3 undescribed bee species, and 21 state bee records for our southern counties. And that those numbers could go up with more study. So what I want to do is just pull out some interesting things from this large project, and I'll start with a project that was developed by this student, Jake Chazowski, who was supported with the NSF Superb grant, and who went on to earn a master's degree in a multi-institutional master's program in Europe, and then now is working on a PhD at Harvard. And so he did a focused study of bees that were collected from 2 limestone glades in Southern Illinois, and that's in preparation to be published somewhere. So he looked at just the bees at these limestone glades, and this is because a lot of solitary native bee. One of the reasons to do this is number one, you find interesting plants in glades and limestone glades, and it also provides a kind of unique or different habitat from most of the area around these areas. And so this could translate into unique nesting opportunities for particular kinds of solitary ground nesting bees. So his study sites, the places that he was focused on were Cave Creek Glade and Lafarge Limestone Glade, and these are indicated on the map. And again, these are areas surrounded by mostly closed canopy forests, but what do you notice here? These are open, they're full sun, and this is the kind of place you want to go to find a lot of bee diversity. Bees like arid areas, full sun, and particular plants. So these 2 areas they're considered high quality glade communities. A couple of interesting things. As it turned out, and I'm not saying there's no non-native plants at Cave Creek, well, anywhere really, there's almost anywhere it's going to have at least a few, but none of them showed up in our study. So in terms of things that were in flower that we collected visitors from, we ended up with all native plants at Cave Creek. That was not the case at Lafarge, where there is high plant diversity, and there are a lot of native plants there, but there's also sort of this entourage of abundant non-natives, invasive type plants. And one thing that shows up in a number of different studies that I'm going to be talking about is an especially large impact of non-native legumes. These tend to have flowers that are adapted for bee pollination, so they attract bees, and that is how they can have a large effect on communities. So I'll show you some networks, and these are frequently just called pollination networks. Technically, what most of what I'm going to show you are visitation networks, which means that you have an assemblage of plants and pollinators, and the lines connecting these indicate an observed interaction. Basically, a particular pollinator species collected from a particular plant species becomes one data point that makes up these lines. And so here you have a variety of plants. Some of them are visited, like the top plant looks like it's visited by almost everything, all of these pollinators. And you can also see that at least a couple of the pollinators only visited one kind of plant, which would be the scenario for specialist bees. And so this is a way that you can sort of numerically analyze these relationships, and you can do calculate some metrics of these networks, which I'm not really going to get into a lot. Maybe I think one slide here, I'll show some of these, but it gives you a way to measure things like the complexity of this particular community, the number of connections versus the maximum number of connections there could be. The maximum number comes from a scenario where every participant visits everybody on the other side. Every plant gets visited by every pollinator, and every pollinator visits every plant. That would be a maximum number of connections. Oh, sorry, connectants, actually. The number of connections observed relative to the maximum possible is more what I just described. It allows you to look at whether connections are clustered on certain species, and this could allow you to look at focal species, for example, dominant floral resources, honeybees, and what kind of effect they're having, or non-native plants, or maybe a rare or declining species. Nestedness is a common characteristic in pollination networks, and it arises from the fact that in pollination, specializations are almost always one way. If you have a specialist bee that only collects pollen from one kind of plant, inevitably that plant is going to be a generalist that's visited not only by the specialist, but maybe other specialists and generalist pollinators as well. And that leads to a measurable quality in these pollination networks called nestedness. You can even use like ecological diversity indices, like the Shannon diversity, instead of applying that to the species that are present, you apply it to the interactions that you've observed there. So we call this Shannon diversity of links. And so this is a measure of how many different kinds of interactions there are and how evenly they are dispersed in terms of all of the connections that were recorded. So what did he find at Cave Creek? These were some of the most abundant pollinators. Got an andrena, a mining bee, one of the metallic green sweat bees, and then some important plants in that are some of these shown here, including Retibita pinnata, which is across all of everything that we collected during these years. This is what we collected. This had the most connections, the most links. It's abundant, and a lot of different things go to it. So what you're looking at over here on the network, which I wasn't really showing on that simpler one, that schematic that I had, is that the thickness of the bars for each participating species and the thickness of the connection going towards the other side is it is a factor of how frequently that interaction was observed. So like that big fat bar going from Agochlorella aurata to Retibita pinnata is a whole bunch of those bees that were collected from that plant. And a very skinny one means that just a few of those bees went to some other plant. This is some of the common species that were collected at Lafarge. And here there are more different kinds of interactions here and more different connections than I think that's evident by the busier network image. You can also notice that some of the non-native plants, Mellotus alba, Mellotus officinalis, where's maybe that's I'm not saying anyway, I shouldn't try to find things on a big graph that I haven't pointed out with some kind of figure. Anyway, there was definitely non-native plant species that were showing up in these as hosts for these bees. Comparison of some numbers here, just looking at numbers of bee species and numbers of floral species. There were more at Lafarge, limestone glade. Some of these network metrics, for example, the Shannon diversity of links indicated higher diversity at Lafarge. Nestedness is a weird measurement. The smaller the number is, the more nested it is. And again, we expect pollination networks to have this nestedness quality. There was more at Lafarge, and also some other measures that are different ways of measuring redundancy and robustness of the community that relate to the number of connections that there are. Were all higher at Lafarge, and this was kind of not expected because this area has a history of mining and the higher presence of non-native plants. It's very likely that non-native plants, particularly if they're co-occurring with a lot of different kinds of native plants, can play important roles in the network and in terms of supporting pollinators, for example. Or maybe and something that's hard to see, has it changed anything? What has it changed that maybe affects native plants? And that's a pretty big question that we weren't out to answer, really. Okay, whoops. What happened? Another little thing to carve out of this is a quick look at floral associations of the declining bumblebee. And this was some work done by Gabriella Burkett, who was also involved in the Superb training grant. And this work is in preparation. So she looked at Bombus pennsylvanicus, the American bumblebee, and this is the declining species. It's actually under consideration for federal listing. When that will happen, if it does, I don't know, but it has undergone a massive decrease in abundance in certain parts of its range, and also its range seems to be shrinking. Most notably, it's being lost in the Northeast and upper Midwest. It still is one of our most common bumblebees here. So whatever's happening to it might be specifically things in the sort of outside of the green area, I guess. So she pulled all of the inventoried specimens of this bumblebee, and also she began some pollen analyses to determine exactly what pollen hosts they were visiting by sampling the corbiculate pollen of the workers. And I'm not going to be talking about that because it didn't get very far, but she did find some interesting things about its floral hosts. And so before I show you what we found, I'll show you a little bit about some stuff, some information on this bee from the literature. So there was a study that came out just a few years ago that found that the American bumblebee seems to depend on fewer host plants than do most other bumblebees. Bumblebees we usually think of them as being sort of super generalists. able to go to just about any kind of plant that's available, but it seems that Bombus pennsylvanicus mostly confines itself to a small number of native plants that include wild bergamot, butterfly milkweed, various silphiums. Uh, uh, well, I'll show you some of the ones that some more plants that we collected it from in the next slide. But they also reported that this bumblebee has strong preferences for certain non-native plants, including red clover and including, I think, sweet clover might be among those. And the significance of this is not as poorly understood. Its attraction to these plants might be driving its decline in some way, or it might be allowing it to persist in more degraded habitats. So the interesting thing is that she looked at about 120 specimens that came from ag fields and what we call semi-natural sites, which is just our way of saying something that's not particularly being immediately impacted by agriculture or urban development or something like that, but is probably also not pristine, I guess. We call it natural/semi-natural sites. And she looked at the floral associations of these and found, unsurprisingly, that in the ag fields, we found this on the clovers that had been planted as cover crops on these fields. And so this isn't all that surprising. It's also worth noting that those three species of trifolium were there for an experiment that was part of this research program. So it's not surprising that we found bees on them. There was also a fairly short list of native plants that either showed up in fallow ag fields or around the edges of them that we found this bumblebee on. But compared to the semi-natural sites, it was a shorter list of host plants, whereas we have more native plants and also some of the same non-natives that have been reported in the literature as being attractive to this particular bumblebee. And that included trifolium as well as some Dipsacus species. Okay, this is Carmen Burkett. She was a master's student in my lab and finished her project in 2023 and is also off now getting a PhD in Texas. For her master's work, she looked at diversity, floral associations, and pollen carriage, and role as pollinators in the Lepidoptera that were collected in our inventory. And I'm also going to be presenting some of her work that compared these natural and semi-natural areas to agricultural sites. She addressed an understudied question: how important are Lepidoptera as pollinators? And I think that the general public fully embraces butterflies as important pollinators. But if you talk to a lot of pollination ecologists, they're kind of like, maybe not. There's some studies that show that they don't transfer as much pollen as bees, for example, or as hummingbirds. But they also, because of the way that they forage, they may move pollen longer distances, and that might, in terms of importance, make up for the fact that they may not transfer as much pollen. So I'll be telling you how Carmen addressed that. By the way, this is one of the ag fields that's planted with crimson clover cover crop And this was the subject of Casey Bryan's master's thesis. She's published part of that. There's so much more that we can publish from this. I better get busy on that. So Carmen also did a lot of the identification. She wasn't around for the collection, the early years of the inventory, but she identified a lot of the butterflies that we had collected. She identified 139 lepidopterans, which were visiting over 200 floral hosts. These were some of the most common butterflies that we collected. Orange sulfur, silver-spotted skipper, the snowberry clearwing. We don't have a lot of hawk moths because we weren't collecting at night or evening or very early mornings. Another skipper, spicebush swallowtail were all frequent visitors that we collected. Really didn't have a whole lot in terms of non-native lepidopterans, but there was this guy that showed up every once in a while. This is a Ailanthus webworm moth. It hitched a ride on Tree of Heaven when it came over here, and it's a non-native associate of that. And it's probably not a pollinator. It's probably just sort of a nectar feeder. I don't know what I did here. I'm going the wrong way. Shoot. I don't know how I did that. Okay. She did find quite a few non-native plants that were hosts for butterflies. And again, we've sort of got this notorious gang of legumes and their criminal leader that they hang out with, I guess, autumn olive. So this is a really terrible invasive. The other one, I don't know that we really know exactly how much these legumes are impacting pollination networks. She looked at pollen carriage. So she scanned these specimens under a microscope and qualitatively estimated the amount of pollen they carried on different parts of their body and came up with a way of an overall score of how much pollen they carried, which we're using as one kind of a measurement of their potential as pollinators. So just because something's visiting a flower and taking resources from it, nectar or pollen for their own consumption or brood or whatever, doesn't necessarily mean they're carrying out pollination. Now, truly testing efficacy of every species as a pollinator for every plant would take forever and many, many people. It's labor intensive. So this is a sort of a quick way to assess this at some level. It's imperfect, but it's really more than has frequently been done. She scanned over 1,000 specimens. So I did this again. Why is this doing this? And focused on, she found that 269 of those were carrying appreciable amounts of pollen, with most of it carried on the thorax, head, wings, and legs. And this was this by family. So you have some butterfly and a few moth families along the x-axis, and then the percent of them that carried pollen. And so you can see Papillionidae. These are large charismatic butterflies that have a lot of area to carry pollen, and they're probably the best pollinators of the ones that we looked at. And so this included the swallowtails and more swallowtails. Sorry, all the swallowtails. We really only had the snowberry clearwing in terms of Sphingidae, the hawk moths or hummingbird moths or sphinx moths, whatever you want to call them. Most of them are not super active diurnally, but that one is, and it did carry a lot of pollen. And then we see a kind of a drastic drop off here where the nymphalid or brush-footed butterflies had lower percentages carrying pollen. And some of the skippers here, they also were quite low. So in terms of that question, are butterflies good pollinators? The answer is probably the same as it is for any large taxonomic group. Well, some of them are, and some of them not so much. She also did some some visitation networks. And here I'm showing you just the agricultural fields, and we'll compare that to the natural and semi-natural areas. So here from the ag fields, we identified 52 host plants being visited by 64 different lepidopterans. And again, some of the major players, this because orientation, I just flipped it, but the plants are on top, the pollinators are down here, and I've listed the ones with the highest strength scores, which is mostly a matter of abundance in their connection in the network. And so some of those wide bars for the plants, those trifolium, weedy things that show up quickly in ag fields. So I think you can just see without reading all of the taxon labels that the natural and semi-natural areas have many more interactions and many more species. So 189 host plants being visited by 77 different species of Lepidoptera here. And again, we have some things like Trifolium pratense showing up, but we also have some good natives here, like mountain mint and ironweed and vervain. So here's where she did something that hasn't been done a whole lot. She compared a visitation network based just on visitation to how this looks when you only include the ones that carried at least some pollen. So visitation networks can tell you about animal foraging, but they're not really telling you a whole lot about pollination because not every visitor is a pollinator. Pollination networks are needed if you want to say something about plant reproduction, how that's being facilitated. And one caveat I'll say is we didn't identify individual pollen types on specimens, although we have those samples now and we can do that moving forward. So this is looking at the same, I've just flipped them vertically now, the visitation network for the ag fields that I just showed you. And here's what it looks like when all of the zero pollen carrying lepidopterans are pulled out, and the host plants that have no visits from anything carrying pollen fall out, and you have many fewer players now in this system. So it drops to 17 lepidopterans visiting 16 plants and potentially pollinating 16 different plant species. And too much information, too much information. These are going to be mostly the species in those families that we just went over that were pretty good at carrying pollen. And now here, same thing for the natural and semi-natural areas where you have this very busy network, and it gets thinned out. Oops, I don't have species names, but again, lots of the things drop out when you're looking at the pollination, specifically at pollinators only or potential pollinators only. Okay, just a very quick bit about syrphids because we have some similar information. This is Jake again. I went on a vacation. When I came back, he had done this. So, and I literally dotted a few I's on the paper and we published it. So he did something very similar to what the results from Carmen's study that I just showed you, but with syrphids. Syrphids are a family. All of the adults are nectar feeders from flowers. The larvae do different things ecologically to earn a living. And we had a lot of them. He identified almost 1,500 syrphid specimens and found 69 species, including this, no, not this one. This is one of the rare ones that was highlighted in the paper, and we also found an undescribed species. On 157 floral associates, lots of different interactions. And he also looked at pollen carriage and did something very similar. So here again, we have the different syrphid flies on the x-axis and a weighted score that scores how much pollen was on their bodies. And so the second bar there, Apis mellifera, is a honeybee, and he wanted to show how much pollen these carried relative to something that a lot of people would consider the gold standard in being a pollinator, a fuzzy, large-bodied bee. And so as you can see, some of these syrphids carried about as much or maybe even more pollen than a honeybee, and others carried very little. So again, that sort of, I think, widespread lore that flies are really great pollinators, and this is something that bee biologists are going to go, are they really though? But now we have some information that again says some of them are probably very effective pollinators and others less so, may mostly be rather benign feeders. And I think I'm out of time, and I didn't get to the the military training sites, but I would be glad to talk about those over our coffee break if anybody's interested. So I'm going to just speed ahead and end with my pretty pictures last time, and then acknowledgments of funders and collaborators and other people who have helped in various aspects of the project. And I'll take however many questions I have time to take, if you have any.